I'm Ali Obedi, serving UMaine as Associate Vice President for Research and Director of the Center for Undergraduate Research. We are excited to host the 12th Annual Cougar Showcase alongside the 25th Annual Graduate Expo for the sixth time jointly as the UMaine Student Symposium. We are excited to see several projects submitted by faculty and students from our regional campus, UMaine Machias, for the first time. I would like to take this opportunity to congratulate all the student presenters here today for a job well done. The depth and breadth of the research and creative activities display is a testament to your hard work. We are in unprecedented times and are hosting our second virtual symposium. This change brought more challenges with it, which you all rose to the occasion and showed even more strength and perseverance, breaking down the symposium submission record once again. I would like to invite guests to take this opportunity to interact with the student presenters to see how the University of Maine and the University of Maine at Machias are impacting the state's workforce and economic development through research, education, and outreach. If you're a new student or have attended the symposium to learn more about research enterprise at UMaine, we welcome you and invite you to use this opportunity to explore the research projects from a variety of disciplines at the UMaine Student Symposium today. Our students that engage in research opportunities are an integral part of our authentic research communities and have remarkable impact on our research programs. They contribute to the generation of new knowledge, advance their respective fields, and ultimately have tremendous success in their future professional careers, due in part to these experiences. If you're interested in research, we invite you to become a part of this vibrant community. I would like to start the opening ceremony by introducing our Vice President for Research and Dean of the Graduate School, Dr. Kodi Varahramian, to make opening remarks.
Thank you, Dr. Abedi. I'm delighted to be joining everyone this morning for the University of Maine 2021 Student Symposium. This annual event provides an opportunity for our students to showcase their research and creative work. As will be demonstrated today, our students contribute to the discovery and advancement of new knowledge that makes a direct difference to the people of Maine and beyond. Moreover, the University of Maine, as the state's public research university, is uniquely positioned to provide our students the opportunity to receive invaluable experiential learning through direct exposure to research and discovery. I also would like to acknowledge that today's event is the culmination of the annual Maine Impact Week, which is intended to provide an opportunity for the public to gain awareness about the latest efforts by Maine's Research University in contributing to the social and economic advancement of Maine and beyond. Finally, I would like to take this opportunity to acknowledge and express my deepest appreciation to the hundreds of students, faculty, and staff who have contributed to the realization of today's symposium. Thank you very much for all the good work. And now I ask Dr. Abedi to please take us to the next portion of the program.
Thank you, Vice President Varahramian. I would like to thank all faculty mentors for supporting a student's research. I would also like to thank the UMSS Executive Committee, the Cougar Office, Graduate Student Government, and Undergraduate Student Government for all their tireless efforts to make this symposium possible. It is now my great pleasure to introduce the President of Graduate Student Government at the University of Maine, Mrs. Saman Zareh, to go over the UMSS 21 program for us.
Thank you, Professor Abedi. And hello, everyone. Today, despite the challenges of the COVID-19 pandemic, over 355 projects presented by 1,200 authors and co-authors have showcased their work during the UMSS 21 in 10 technical categories covering arts, humanities, engineering, education, natural, physical, and social sciences, health, life, and biomedical. And for the first time, we have included an interdisciplinary research category to promote and celebrate teamwork from multiple colleges and departments solving a complex problem. All presentations are virtual and posted online with accessibility features. Each presentation is judged by 3 judges and scored according to the published Symposium rubric. In addition to the technical sessions, next we have a panel of speakers to discuss the COVID response from business leaders in Maine. We have a great keynote speaker, Dr. Nirav Shah, Director of the Maine CDC at 11:30 a.m., followed by the award ceremony at 2:30 p.m., which you don't want to miss. If you have any questions about any of today's events or how to find them, please chat with us at our live help desk at umaine.edu/UMSS. We have teammates standing by to answer all your questions. I would like to thank our sponsors who make this event possible with their generous support. 2021 sponsors are Student Government, Graduate Student Government Center for Undergraduate Research, and Office of the Vice President for Research and Dean of Graduate School as our host. Our diamond sponsor is Texas Instruments, and our platinum sponsor is Maine Technology Institute. Our gold sponsors are Activus Diagnostics, IEEE Maine Section, and Northland Light Health. And our silver sponsors are Allied Engineering, the Jackson Lab, Landry French Construction, President John Fernie Mundy and Richard Mundy, ND Paper, Power Engineers, SMRT Architects and Engineers, St. Joseph Healthcare, TRC Companies Inc., University Credit Union, and Upstart Center for Entrepreneurship. Thank you, everyone. Now we would like to introduce our panel of speakers on COVID response from business leaders in Maine. From Abbott Laboratories, Norman Moore, Director of Scientific Affairs. From Jackson Lab, Nadia Rosenthal, Scientific Director. From Puritan Medical Products, Mehdi Karimchi, Vice President of Scientific Affairs. Timothy Template, Executive Vice President of Global Sales. Virginia Template, Marketing Manager. And Derek McKinney, Engineer. Please be sure to use our Q&A feature here in the Zoom webinar if you have any questions for our panelists today. I would like to thank our panelists to join us for today. I think I saw all of them attending. So I would like to start with our first question today, which is, can you tell us a little bit about your organization and how it has been involved in the response to the COVID-19 pandemic? I would like to go with the order that I introduced our panelists. So I would start with Norman. Norman, thank you.
Well, thank you. I think people need to understand Maine was actually one of the first real states to do a lot of respiratory testing in general. 20 years ago, a little company called Binex actually developed one of the first big influenza tests that became the worldwide test for influenza. And so having this Maine company that then got bought by Abbott, having the ability to already do influenza and respiratory syncytial virus, we had that base knowledge. So when COVID hit, the company leaned on us to come up with those tests. So we have now at that point, developed a rapid molecular test, because the whole issue is we don't want people going into a hospital. We want them tested as close as they can so they're not transmitting it to other people. So we built a giant brand new factory, and we've really put a lot of effort in so that we're making, 50 million tests a month, which is unheard of. So it's really been our pleasure and it's a lot of exhaustion with all the people here to really do what we need to do and work with other vendors like Puritan to really stem this, because you can't hold back the pandemic without proper testing to isolate those people. So it has been a long road, but it is amazing what Maine has done.
Thank you. Next, Nadia. Hello.
Yeah, hello, good morning.
you for
I'm
joining us.
I'm coming in from the Jackson Laboratory. At the JAX, we have several initiatives to address this pandemic. The first was one that we started back in March of 2020, when the pandemic was clearly underway, and we realized that one of the major issues facing the field was the lack of a decent preclinical model for testing of the virus. Mice, which of course is our bread and butter down here at the Jackson Laboratory, where we have 2 million mice, and I guess it's about 11,000 different strains. And we are a nonprofit organization that distributes these mice worldwide. We have the opportunity to work fast to try to develop a good model. Well, it turns out it's hard to develop a good model for COVID-19. Everybody knows from the news how many different ways patients suffer and survive this infection, ranging from really severe responses to asymptomatic. And there's absolutely no way at this point to predict what is going to be your response to this virus. So we can't really imagine one model that will fit every possible patient out there. And so I took it on myself to develop a whole slew of models, a panel of models in the mouse that would hopefully be able to model or mimic the responses of the patient population globally. And to make a very long story short, we teamed up with the Rocky Mountain Laboratories in Montana, which is part of the NIH and does the really high containment infectious disease research for the NIH, to look at a number of models we developed where each mouse had a different genetic background. And by testing these mice with a cross to a known transgenic sequence that conferred susceptibility to this virus via a human receptor, we were able to generate a number of mice, all of which could be infected, but several of which showed asymptomatic responses, others showed severe responses. And we have now characterized these animals and are basically able to offer the community within a short period of time a real a variety of different ways to study the virus. So that's one early response. Subsequently, one of our faculty, Ryan Toohey, decided to contribute to Maine's response to the pandemic by systematically sequencing the actual genomes of the viruses that were being isolated in Maine. And as we know now, there are a series of variants that are showing up around the world, and by definition, they're arriving in Maine. This has been, I think, a real key component of bringing the pandemic under control in this state, and these surveillance efforts are ongoing. We also are developing a number of tests to make sure that people's mice don't get infected in case the variants shift to be able to infect them. So that's enough for me for now.
Thank you, Nadia. Okay, so from the Puritan, our first panelist would be Sir Mehdi Karamchi. Can you hear us, Dr. Karamchi?
Yes, good morning.
Good morning. So the question is, can you tell us about your organization and how it has been involved in the response to the COVID-19 pandemic?
Well, Puritan Medical Product is primarily in the field of specimen collection, preservation, and transport. So since beginning of the pandemic, obviously everybody realized that there was shortage of swab and preservative. So Puritan has been involved from the beginning in expanding, and we added already 2 more facilities in Maine and 1 facility under construction in Tennessee. So we have raised our production and from 50, 60 million initially, now up to around 200 over 200 million swab a month. So we have been able to, over a short period of time, to increase our output in order to be able to provide swab and preservative unit, sample collection unit to our customer and to the different hospitals and unit in order to be able to collect sample for use in pandemic.
Thank you, Dr. Karimchi. Also from Puritan, we have Virginia Templet and Derek McKinney. Hello and welcome. Would you like to add anything before we move to our second question?
Yeah, Derek McKinney here, Director of Engineering. Just to give a little insight on our expansion efforts. We have been producing diagnostic specimen collection devices for over 50 years now. And we have many developed technologies in-house that were, we'll say tailored to lower volume production. We've not traditionally been in a situation to produce hundreds of millions of swabs per month. So as part of our COVID response, we had to sort of change our philosophy around manufacturing, move to fully integrated and automated lines, going from lines running at 100 pieces per minute to lines running at 600 pieces per minute. So it was quite a massive technological advancement in a very short time, combined with at the same time building facilities from the ground up. The first one was about 100,000 square feet, and it was completed in 8 weeks and brought online about 40 million foam swabs per month, scaling to 90 million foam swabs per month. Following that facility, we focused on nasal pharyngeal swab production, flock swab production, and built the second facility capable of producing 50 million nasal pharyngeal swabs per month using similar automation technologies. So we've greatly increased our capacity in a very short time. And moving forward with Tennessee, we'll be looking to move from that facility, scale up to around 200 million anterior nasal swabs per month. So that's going to be coming online through the summer, and we'll be, there's a lot of work left in front of us, but we've got a great team, and we're here to provide solutions.
Great, thank you, Derek. Okay, so our second question is, how has the COVID-19 pandemic impacted your company and changed your goals? So let's start with Norman.
It would be easier to say what hasn't changed. I mean, unfortunately, literally, we had the scientists who basically had to work 24 hours a day. We would literally have, half working 12 to hand other things over to another group that was working 12 hours. So I can't say enough about the scientists that were willing to give all of their time and effort and blood and sweat during those early hours. But then again, we also had our engineers, again, trying to build a whole new factory before we even had a test. So between it was an amazing amount of cooperation. It was an amazing amount where we were relying on the bigger Abbott to bring in their resources to help us, because again, to go from so many million a month to 50 million tests in 2 different formats, we literally built, so I'm just so proud of how many people, and I guess that's why I'm kind of stumbling over the words, is because it became such a great team effort between scientists, between engineers, between everybody else to try to get these tests done in a short time to our suppliers. So nothing is the same. Anybody that wasn't in production got kicked out. In my office now and is in another little building because that whole area had to be made production. So we've all had to sacrifice for the better good of the country. So I'll kind of leave it at that.
Thank you. Nadia?
I think I echo Norman's sort of pride and amazement in how The staff at JAX, like at Abbott, has really stepped up for a real challenge here. You know, mice don't get COVID, like I just said, but they still need to be fed and watered, and so they need humans to do that. And that meant that approximately 1,000 employees that take care of our mice in Maine couldn't really stay home. Especially in the early part of our response, when testing really available the way it is now, where we're all tested weekly, it was, I think, a real sort of act of bravery. It definitely changed our work pattern because those of us who weren't actively in the laboratory elected to go home and work from home. So that includes me. I only go in from time to time to make sure everything's okay. But in general, the administration is staying out of the way of the people who need to be there for the mice. And now slowly we're being able to open as vaccination increases and bring the scientists back in. We went down to 25% of our research capacity during the early stages, just to make sure that we didn't endanger those employees who did have to arrive. I'm very proud to say that we've had a minimum of infections and none that have originated out of our campus. They've all been people bringing it to the campus, and we've been able to contain that. So we didn't have community spread. You know, it's been a remarkable year for everybody, and I'm just particularly proud of those of our JAX employees and managers who made it possible for us to keep supplying the world with mice, even during this really stressful time.
Thank you. Before we move to Dr. Kramsky, I just want to remind our audience that they can use the Q&A. Marzola, I've Zoom here to ask their questions. Dr. Kramsky, please go on.
Well, Puritan was in the same situation as others. We all of a sudden found ourselves in a situation that our customer, Pfizer, Roche, Abbott, and all of them are looking for a specimen collection and transport system. All of a sudden, we found ourselves involved with Pfizer in their vaccine development process because they needed millions of specimen collection in order to go ahead with their own investigation. Abbott, the same thing, and we were working with Roche and other major pharmaceutical and diagnostic laboratories to cater to them in order for them to be able to deliver their tasks. So we went from 40 hours a week production to 70 hours a week production 7 days. And this has been going on for a year now, at least in the area of a specimen collection and transport system. And I believe it was the same with swab production and scale-up processes. And I think Derek can speak to that as well.
Thank you, Derek.
Yeah, and I think we all echo Norman's comments. I mean, it really speaks to the great workforce here in the state of Maine and what we're capable of. We were working because we were still coming out of a flu season, so we were still working some overtime, some extended hours at the start of COVID, and we moved to 7 days a week, 2 shifts in a response to COVID. And I mean, Puritan was a small company pre-COVID, right? One manufacturing facility, a very small group of people. And it was, none of us had any experience in building a second facility in 8 weeks, let alone a normal 18-month timeline. So it was a great effort that took a great team of very motivated people that really went above and beyond and committed to, 12, 14, and 16-hour days for weeks and weeks and weeks on end. And it was really something amazing to be a part of, and truly shows, what a small group of motivated people are capable of doing.
Thank you, Derek. Our next question comes from our audience. I think it's been a question for many. It's about the Johnson & Johnson vaccine. And the question is, will your goals change now because of the Johnson & Johnson blood clot and pause in the distribution? Because students are now worried, according to our audience, about taking the vaccine. Should we be worried about the vaccination rate in Maine because of this?
I get into trouble every time I post on Facebook that if somebody says something, and trying to, it is difficult. 30 years as an infectious disease scientist, for many people, that's a stack up against the 8 Facebook posts that they've read that now make them a better expert than my 30 years. And I think we're all kind of struggling with that. So it is difficult. But the thing I want to really warn people, and it's more about messaging, is that we have worked so hard on these vaccines. The worst thing we can do is let up, have a bunch of people unvaccinated, and then the unique variant comes from that group, and we start this whole thing over again if we really don't get this herd immunity built up. So what I try to relay to those people is, yes, you may not be the biggest risk group that's going to get the sickest, but we need to not let up right now, because I want to go back to no masks. I want to go back to 4th of July and hanging out with people. And that means that we have to do what we can. So, and I'll leave it there, but I can't help myself when somebody posts something on Facebook and then I get totally blasted for being part of the scientists. I'm not used to scientists being the bad guys in all this, but we've had to face a little bit of that. So that's my messaging, is please don't let up. I want to get out of this. I'm sick of wearing masks myself.
Thank you, Norman. Nadia, do you want to add anything?
Well, I think, you just have to listen to the facts and make your own decision. The fact of the matter is that your chances of getting a blood clot from COVID-19 are hugely increased over any chance of getting a clot from the Johnson & Johnson staff. We're looking at 6 women out of 6 million people, or it's something like that. I can't remember the exact numbers. You have to be able to do a little bit of statistics to figure this out, but it doesn't take a mathematician, I'm not one, to look at your risks. The fact of the matter is that you're much more likely to get in a car accident than to get some problem from this vaccine. And given that you're also much more likely to get COVID without the vaccine than with the vaccine, I don't know, I'd take it. My husband's had it. He's just walking around right behind me. Sorry about that.
Thank you. Dr. Kamchi or Derek?
I'm in the same line. I believe the vaccination should take place, and even if it can be faster, because we won't allow the virus to further mutate and create more variants. So it should happen, and it should happen faster for the sake of public safety.
Thank you. Our next question is about the virus and the pandemic, as you know, are changing forever, probably. But how will you plan for the mutation rates and variants that continue to dominate? Norman? Okay.
So you're asking, I will say this. I went into virology because it's not hard facts. We really, there's a lot of guessing, and anybody that says they know exactly what's going to happen is probably lying, or a little bit, we don't fully know. So how do we plan? Again, the planning is to actually, unfortunately, keep up with what we're doing. Wear the mask and drive it down to that herd immunity. We've got to get as many people vaccinated so that we can. I mean, it looks like it's mutating very fast. And one of the reasons is because we've had so many vectors, so many people getting infected. So the more people are infected, the more likely it is to mutate. So it is working with a global community to make sure that we get this as safe as possible by getting this. So, viruses, bacteria don't recognize borders. So whatever happens in one part of the world does affect others. So it is a global community effort.
Thank you. Nadia?
the variants are going to be with us forever. We know from previous pandemics that viruses mutate continuously. The reason that they have become so prominent in this particular case is because there are so many people infected, so it has a huge field to work in. And so just because of evolution, its chances of mutating to something that is more infectious is very high. From a virology standpoint, I used to be a virologist, the trend for viral mutation is away from serious infectious severity. Why? Because viruses need living hosts, not dead hosts. So if you think about it from an evolutionary standpoint, the virus is better off giving you flu than killing you. The issue is simply, at this point, how do we contain the virus, which is already a bad thing? And I think that one of the great triumphs, really, of the incredible speed at which these vaccination protocols have been developed and the vaccines have come out is that among these vaccines are vaccines that are RNA-based, which means they are made out of nucleic acid, which is much, much easier to engineer than protein. So if you can simply tweak the RNA in either the Moderna or the Pfizer virus, there's a chance that you can more rapidly tune and tailor the new vaccine to a new variant in a short period of time. I mean, that's the company line. But of course, as Norman knows, it's one thing to design a vaccine. It's another thing to distribute it. So we're not out of the woods. Keep your masks on. Follow Dr. Shah's advice and stay safe.
Thank you. Dr. Karimchi, I just wanted to make the question a little bit broader. So not just about this virus and its variants, but about other outbreaks in the future or public health crises that we experience. So how is your organization planning for the future of biomedical and health sciences?
I guess what we have done in the past year, obviously we have expanded our capacity. We have gone from one plant, and the hope is by the end of this year we will operating 4 different plants, 3 in Maine and 1 in Tennessee. And I don't have the exact number. Perhaps Derek can help in that, but I believe we have now enough capacity that if there would be another pandemic, we will not be in a situation that we were a year ago.
Thank you. Derek?
Yeah, that's been our goal, is to build our specimen collection lines to sufficient capacity for rapid rollout across the country. And I think, with partnering with Abbott as well, to develop, rapid testing that's easily accessible really has been one of the biggest goals of the last 12 months. And I think going forward, rapid testing, at-home testing is going to remain a part of everyday life to some extent. And that's what we're preparing for.
Thank you. Okay, and another question from the audience, which is like we expected, is about vaccine safety again. So the question is, how might you approach discussing vaccine safety with a loved one who is vaccine hesitant or anti-vaccination?
Probably trying to put it in a bit of perspective. I mean, I think the birth control pill, gives more blood clots than the Johnson & Johnson vaccine. So when people, again, like Nadia said, put it in broader context, I think it really kind of helps understand this. But the other thing I kind of try to point out is, we have a thing called long haulers now where, we have these people that really aren't fully recovering. And as a scientist, I don't know the long-term implications of what's going to be happening with the heart. I mean, we're hearing COVID toes and all of these other unique things. And so when you're dealing with a piece of RNA that's expressing a part of a protein versus I don't really know what this virus is going to do to people long-term, are we going to potentially have a loss of smell later on as those cells that we lost them and regenerated, maybe they go back again? The risk is far, far, far greater with a virus that is affecting our cells, and we have that long-term impact. So that's kind of the messaging I've been trying to give people, is that is a far bigger risk. And again, with the blood clots, just trying to measure that to other things, but putting into context, like Nadia said, has been not perfect, but it helps. We have to make sure they understand these big odds.
Thank you. Nadia?
Yeah. First of all, I'm sorry about the background noise. There's a fire in the room, and there's a log on it that's crackling. So just think of it as cozy rather than stapling in the background. The way that I deal with this question is in very similar way to the way Norman does, which is to point out that life is full of risks, that the companies who are generating these viruses are under enormous pressure to get them out, but are under even more enormous pressure not to generate a reagent that's going to do harm. So the fact that we are not FDA approved yet, which is one of the arguments I often get, shouldn't really keep people from being willing to take the risk, take the virus. It's an incredibly small risk that take the vaccine. It's an incredibly small risk that you will have a bad reaction to this vaccine. Vaccines are not designed to be toxic. I can tell you, I've had the Moderna second shot, and I fell asleep in 2 meetings yesterday, but as you can see, I'm back to normal. And it wasn't a bad thing at all. And I know what's going on in my body, so it helps. As a scientist, I'm not scared. But frankly, what you're doing is giving your body a chance, in case it does encounter the virus, to clamp down on it, just like it does after a flu vaccine or any other vaccine you may have had in your life, including polio and others. So I think there has been an anti-vaccination sentiment that has grown, which is totally unwarranted, based on data that is faulty, and doesn't actually reflect the extraordinary numbers, millions of numbers of people who have been vaccinated across the world and are now showing that where populations are showing extraordinary reductions in infection rates. So we're doing what we need to do to avoid those long-term consequences you heard about from Norman, which are the really worrisome part for me, and would be the worrisome part from anyone in my family that got COVID and maybe didn't even know it, but later on might have some of these very worrying downstream effects. And it doesn't help to be young, to be immune to those long-term effects. Just as many young people are experiencing them. So for all of you listening, go get vaccinated. Don't be dumb. Listen to Polly Parton.
Thank you. Mehdi?
I feel the same. I think the chance of adverse effect from a flu vaccine is 1 in a million. There's always a risk crossing the street or getting hit by a car. So there's always risk in everything. So, but the risk and hazard associated with it is way, way lower than, just normal. This is the way it is. And I think we all should get vaccinated.
Thank you. And Derek?
Yeah, I agree. I mean, I'm an engineer, so risk assessment and risk management is part of daily life. And I think understanding the risk on your own terms, evaluating the information that's available. As we said, looking at the facts is key, and not getting distracted by some things that might be out and available in the media.
If I can add too, just I forgot this. Vaccines have added over 9 years to the length of life we had in the United States. I mean, that vaccines have suffered from their own success, with not having polio anymore or smallpox. And for people that really don't believe in vaccines, I would suggest you go talk to some of the more elderly people and ask them what happened within the days with polio when their friends all of a sudden woke up and couldn't move their legs. And I think you're going to find that the generation that really didn't have those are going to be some of the better teachers to say, this is what happened in the days before the vaccines. So look to those people that have experienced that. And I think you're going to find a very unique story saying how much they have improved by getting rid of polio in this country. And we've almost got that eradicated in the world.
Thank you. Just before we move to the next question, I just again want to remind our audience that you can use the Q&A feature here in Zoom to ask your question. So the next question about the assistance you received, your organization. Did you receive sufficient assistance from state and federal governments during your rapid response to the COVID crisis, or is there anything else that you wish the state did to help with your operation?
I think it was a bigger partnership than I've seen in all my years. Even for the state of Maine, if we needed a permit to move, things down the highway on the weekends, whatever, we got those permits and we were able to build an entire factory in a very short order. So, there's a lot of thank yous to the state of Maine, and when we talk about the government, every time I called the CDC when something happened and we needed to work with them, they answered that phone in as many hours as they were working. The FDA, I can't say enough about them trying to see how we could work and try to get these tests out. So it was an extraordinary effort, and our government partners really, to me, came through. I was very happy with the state of Maine. I was very happy with the U.S. government in trying to get this done.
Thank you, Norman. Nadia?
I can't say enough good things about both state and federal governments, and I echo everything Norman said. There's been an enormous pulling together. At JAX, we very rapidly not only collaborated with the National Institute of Allergy and Infectious Diseases to get all of our mouse models tested in their facilities out in Montana, which they are still doing free of charge, but they then sent us abundant funds to make sure that we had enough funding to develop these models as rapidly as possible. Likewise, we've been very lucky as scientists to have access to a number of supplements that have been issued by federal agencies to support COVID research. And then finally, the Maine CDC has been a wonderful partner in our attempts to sequence and do the genomic surveillance on the variants as they come through, because we're now getting the UK variant and pretty soon we'll have the others if we don't already. So I can't say enough good things about both state and federal support.
Thank you. Mehdi and Derek?
Yeah, I would say the same thing. We have had a significant amount of help from federal and state government and also regulatory agency. Even this week, we were approached by FDA, just checking to see how we are doing, whether we need any help or if there is anything they can do to speed up the processes. So I can say that in all level, it has been great. Derek can add to that better than I can.
I mean, I think from the very beginning, we had great support from the state of Maine. One issue in the spring we faced was, the potential of power outage, which could affect production. And the state was there, to provide supplemental power generators if needed, working with, coordinating with CMP. I mean, anything we needed from that end, the state of Maine was behind. And then for those that don't know, our expansion efforts have been funded as part of the Defense Production Act. So we've received over $350 million in federal funding towards our capacity expansion, and have really worked with an amazing team in the DPA office. Everything from helping to source long lead time components on equipment to accelerating lead times on air handling equipment. I mean, just across the board, have supported the initiatives and in any way that they can. And it's just a great team and great support to get the country to where we need it to be.
Thank you. Okay, our next question is from our audience. The question is, what inspired each of you to pursue your careers? And what advice would you offer to undergraduates working towards a career in microbiology or medical laboratory sciences?
When I was an undergraduate, I always knew I wanted to be a scientist. And I remember taking, that first microbiology course, and I found myself just going back at nights to do those additional experiments. And if I'm willing to go back at nights to do extra work for a course, you kind of know that that's your career. And I just love the uniqueness. You cannot predict what's going to be happening. Like this pandemic, we don't know. And so I love how much this field changes. So there is, maybe that sounds geeky, that's fine, but there's always been a level of excitement with infectious disease. It is so unique. So I like the constantly evolving field. And of course, we're trying to do here is new technologies. And, how do we find these bacteria and viruses faster and isolate and help things? So it's been a utterly rewarding career choice for me. I am thrilled that I have been an infectious disease scientist. I would do it all over again.
Thank you. Nadia?
Well, I got out of infectious diseases at a very early stage back in the 80s because I was distracted. But I'm delighted that the opportunity has reappeared for me to brush off my virology a bit. I'd say that everything that Norman mentioned is absolutely the way I feel about being a scientist. But I'd also like to point out that capacity to turn left, change your thought, change your ideas, change your pursuits. You don't get that kind of opportunity very often in life. There's always somebody telling you what to do. And of course, being a scientist isn't easy. You have to raise money. You have to make sure that you have enough funds to support the people who are dependent on you. And it isn't always easy to deal with the fact that 90% of the time you're wrong. You have to get used to that. But that's just humbling. And I find that the truth is so much more exciting than the depression of not being right, that it's worth the wait. The other thing I'd like to say about science is people think of science as a kind of solitary pursuit, the crazy scientist in the lab late at night with the bubbling test tubes. That's just not the way it works. The way it works is you're in an incredible community of people who love nothing better than to talk science. They like talking science better than they like talking baseball. I mean, it's really fantastic. So there's the other thing is I've always found, I've worked around the world, and I've always found the interesting part of that is that wherever you are, and no matter what time of day it is, there's somebody who's awake that you can talk to about something that occurs to you. So I've been known to wake up at 3 a.m. with an idea and call my friends in Europe to ask them about it. So it's just, I can't describe how it really even preceded our now sense of global sort of connection with the web. Back when we had to actually write letters or send faxes, we were still crazy and we were still doing it at 3 a.m. So if you like a life of adventure where nothing stays the same, as Norman pointed out, and you have friends at every part of the globe, join us. It's a wonderful career.
Thank you so much. Mehdi?
I feel the same. I believe there is a lot of work to be done, especially in the area where we can use bacteria or viruses and study more genetic aspect of bacteria or viruses, and use that technology in future in sort of curing diseases. We are just at the beginning of this field, and I think we can advance a lot rapidly using microbiology and advanced diagnostics and curing in terms of cancer or other significant diseases. That's a future of the microbiology.
Thank you. Derek?
Yeah, so I mean, I can just say that in working in the medical device field, whether it's, you're making implants or you're making specimen collection devices, it's, unbelievably rewarding because your end goal in everything you do is always focused on the patient. And it's always improving the quality of someone's life. And it's truly one of the most rewarding fields I could ever imagine working in. I mean, I absolutely love my job. It's a joy coming to work every day for Puritan. And it's just great to make a difference every day in the world and every day in someone's life.
Thank you, Derek. Speaking of career and career opportunities, the question that many of our students and attendees may be interested in is, what kinds of opportunities are there within your organization that students could become involved either during or post graduation? Norman?
Pick one. Engineering, quality assurance. We're still begging for quality assurance people. You've got, I can't say enough about our engineers trying to, again, build this place out. Obviously, our scientists, and we've got different types of scientists, because again, just like Nadia said, we've got to work in a community. It is not, scientists don't work in these little solo things. And so by relying on each other, there is a ton. So, we have people doing our clinical trials, and that itself is a phenomenal job of, understanding and making this work to present to the FDA. So infectious diseases will not be going away. We will get a handle on COVID, and it should be in the end a seasonal issue with many other respiratory viruses. But, infectious diseases are here to stay. We've got MRSA, we've got drug-resistant gonorrheas coming out of, other countries. There's plenty of jobs. Infectious diseases don't go away. I'll leave it at that.
Thank you. Nadia, I would also like to add another question to that. Besides career opportunities in your organization, what kind of skills you would encourage students to develop to prepare for such opportunities?
Sure.
Is that for, okay, sorry, go ahead.
Oh, no, go ahead. Please.
No, I think that was for you, sorry.
All right. First of all, I'll just mention a few opportunities at JAX that we constantly have open. Things are a little different this year. We're hoping things will get back to normal. Normally, we have a very active summer student program. We often have undergraduates coming to take on projects that they can work on during the summer. And for those kinds of projects, really what we're looking for is basic analytical skills, a bit of math, some biological background would be very helpful. But really, the best thing that I always look for in a student is enthusiasm and perseverance. It's a question of whether coming into the lab and doing the same thing over and over again because it didn't work the last time is going to drive you nuts or not. So it's a kind of a stubborn mule type of personality that tends to do very well in science. Like, I'm going to get this no matter what. It's certainly how I got through my graduate training. And then, of course, after a graduation, there are a number of opportunities. University of Maine has an active PhD program, which we share with the Jackson Laboratory, which we share with you. The Jackson Laboratory shares with University of Maine. We are inviting students to consider the PhD programs here. We have an incredible variety of areas of research that you might be interested in, from cancer to to aging and Alzheimer's, to my own work in the heart. And now with COVID, we have another opportunity which has opened up, which is not quite ready for prime time yet, but Dr. McGinnis, who's on our panel today, or is certainly on this call, and I have set up a collaboration so that JAX can start working in the new BSL-3 facilities that have opened up at University of Maine, since we don't have any BSL-3 facilities at JAX. And you need a BSL-3 facility is just a high containment facility where viruses of considerable human danger, like SARS-CoV-2, can be effectively and safely used. So in the future, we're really hoping to engage students in the kinds of collaborative work that Melissa and I hope to develop.
Thank you, Nadia. Mehdi?
Yes, we have been actually benefiting from the University of Maine undergraduate program in the past decade or more. We have had several students that have joined us, and there are usually two types of a student. The local one that they join Puritan and they stay here. We have a quality control manager here. We have a production manager. These are graduated from undergraduate program at University of Maine. And we have others that when they graduate, they join us and they gain experience for 2-3 years and they move on, join a Lear or go to other major centers to work. But we have been significantly benefited from that, and their science program would help us. And we offer them opportunity either here to grow with us or to move on, gain some experience and move on either to advance their education or to the other centers.
Thank you. Eric?
Yeah, I mean, we have opportunity across the board in engineering, validation support, manufacturing, process development. QA is a large need, regulatory, as well as operational management. I mean, across the board with the rapid growth, we're still responding to the staffing needs. And now in Tennessee, obviously, we'll be developing a truly remote location to corporate here in Maine. So the needs are near endless at this point. So there's opportunity on top of opportunity here.
Thank you. Our next question is from our audience, and it's about vaccines. So the question is, for people who are fully vaccinated, how soon do you expect it will be necessary to get another shot? Will this become an annual vaccination like flu? And is it reasonable to think that someone who's vaccinated will be okay for a year, or will another shot be needed sooner? Norman?
All right, so this is a guess, and we obviously we haven't had the time yet. So, if I were to take just a wild stab guess, I would say probably 2 to 3 years, but there is some people suggesting that it may be next year, but it's going to be a wait and see on this, which is like every other infectious disease, we have to understand it. So it would not surprise me. And my wild expectation, again, do not take this to the bank or anything, that it's going to be more in the end of a seasonal thing where we really worry about the young and the elderly, just like respiratory syncytial virus and other things. And we take the vaccine again. I take the vaccine to protect my mother. I take the vaccine to protect other people. So that's how I look at it. But I would guess right now every 2 or 3 years in the future. Obviously, let's see what happens with herd immunity, but we'll see.
Thank you. Nadia, what's your expectation?
Well, I'm going to defer to the professional virologist and let Norman speak for this. And I would be obviously very delighted if we made it for 2 or 3 years without the emergence of some variant of this virus that requires a whole new round of testing. again, getting back to vaccination, the best way we can prevent that outcome, namely new viruses popping up all the time, is to get rid of the one we have. Because unless we get rid of the one we have, we won't deplete the pool of viruses that can mutate. So if you lower the number of people with virus right now, you lower the number of viruses out there that can mutate, and the chances of us having a new variant go down. I mean, I'm not saying anything that Dr. Shah hasn't said 1,000 times, but I'm just saying it again. Get vaccinated. It's a really good idea for you and for everyone around you.
Thank you. Dr. Karinci, I would add another question from our audience to that question, besides your expectation about how often we should get the vaccine. It's been told that we won't get over pandemic until we get all the world vaccinated. So the question from our audience is, how can we encourage and facilitate making vaccines available in countries where they're not produced?
That's a great question. I think we cannot basically believe that if we vaccinate everyone in the United States, basically we can get rid of this virus. This is a global problem and it needs global solution. And if this vaccine mutate as rapidly as it is, no matter how fast we immunize people in the United States, the traveling will start and people start traveling. They bring the variant into the United States and this will continue. The key issue is, although we don't have an answer how often we need to be vaccinated or whether it's every year or two or every 3 years, the important thing is to vaccinate in a global scale as soon as possible in order to stop virus from mutating and creating more variant. That's really the goal. We will find out in a year or two how this vaccine will react and how often we need to be vaccinated as the research continues. But one thing we all know for sure that is to vaccinate as quickly as possible in a global scale.
Thank you. Derek, do you want to add anything?
I think Mehdi covered the bases, and I'll leave the rest with the experts.
Thank you. Okay, I think that was the last question from our audience. So I think we should start the closing part of our panel. Norman, Nadia, Mehdi, and Derek, is there anything that you would like to share with our audience before we finish the panel? We'll start with Norman, please.
I think the first thing is for the people that were the front lines, working in the medical establishments, it's a big thank you. I can't say enough for the people that have been working in the hospitals and how tired some people are and the exhaustion and all us going through that. So first of all, my thanks to all those people. And then I guess secondly is, it's still all of us working together. What happens in one group may affect others. So, yes, I've been vaccinated. I got the fortune to be vaccinated because, obviously they want to make sure all of the plants stay open and we don't infect other people, but I'm still going to be wearing my mask and trying to do that so that we, again, keep driving it down to zero. I don't want to let up right now. I want to go back to normal, and that means getting it to the point of herd immunity rather than saying, oh, well, I don't see as many cases right now, let's relax our efforts. So I guess that's my issue is, if you're sick of masks, I am very sick of masks. That means drive it down to the point where we don't need it. And that means don't let up right now.
Thank you, Norman. Nadia?
Yeah, Norman said it all. I'm in great awe of all of the incredible people around the world who have really stepped up to try to help those who got infected and are in real trouble. I'm also very impressed with the way in which numbers of companies have really managed to pivot towards production of vaccines and other antivirals, and all of the engineering that went into getting all those tests all over the world. It's really, it's a good news story, except for the fact that it's a good news story on top of a very bad news story. But I think it's taught us all a lesson about the power of nature. We don't really know how to control it. And it's amazing to me, the little piece of nucleic acid, as someone once called a virus, a little piece of bad news wrapped up in protein can really mess up an entire globe to the point where we are now really not talking about anything else. I'm looking forward to talking about science and the weather. And as you heard from Norman, the best possible way that we can all help is to maintain as much as we can, you know, our natural tendency in Maine to stay away from each other and keep those masks on. I think the lucky part about being in Maine is that there really is a chance that we could get it down to zero. We are the first in a number of ways, and we're very close to the first in our capacity to do the scientific research we need to do to try to maintain vigilance over those variants and try to keep an eye on when they come up and get rid of them as fast as we can, so that we will be a state where we can celebrate the 4th of July together again. And that would be just my best imagined scenario. So I'll leave it at that.
Thank you, Nadia. Mehdi?
Yes, Norman and Nadia said, said it well. And I basically would like to thank all the frontline worker. Keep mask on, keep your distancing until we defeat this enemy.
Thank you, Mehdi. And Derek?
Yeah, I agree. It's a big thanks that goes out to everyone that's been part of the response in the state of Maine and in the country. It's really shown, in the time of in the bad news, as we say, a good story has come out of it. It's very encouraging. And I think now, especially as people are starting to travel more and go back slightly to a little bit more of a revised normal, that we don't lose sight of the end goal here and that we stay focused. And stay cognizant that, we need to continue wearing our masks and just be a little bit patient, and we will get there. So just, stay focused and stay safe.
Thank you. Thank you to all of our panelists today for taking the time to share their experiences during the pandemic. This has been very informative and helpful, and I'm sure that our audience enjoyed it as well.
My name is Melissa McGinnis, and I am the Associate Director of the Center for Undergraduate Research. It is my great pleasure to welcome Dr. Joan Farini-Mundy, the President of the University of Maine, and our regional campus, the University of Maine at Machias. President Farini-Mundy holds a PhD in mathematics from the University of New Hampshire and was a faculty member and associate dean at Michigan State University. Prior to her presidency, she served as the Chief Operating Officer for the National Science Foundation. She is passionate about providing research education experiences for our students and is leading the UMaine System Research and Development Plan. Over this past year, President Farini-Mundy has led us through the challenging navigation of the COVID-19 pandemic as the president of our universities and also as the chair of our UMaine System Science Advisory Board and the Vaccine Planning and Partnership Task Force. Please join me in welcoming President Farini-Mundy to the podium to introduce our keynote speaker.
Thank you so much, Dr. McGinnis. It's a delight to be here, to welcome all of our student participants, and to congratulate you all on the wonderful work you're doing in research and in taking next steps toward the rest of your career. It's my pleasure today to introduce Dr. Nirav Shah, who is director of the Maine Center for Disease Control and Prevention, and he's our keynote speaker for the 2021 University of Maine Student Symposium. And as we were just chatting a little bit, you're going to have a phenomenal chance to ask him questions. He'll welcome hard questions, challenging questions, and you will get thorough, clear, and very, very useful answers. So this is a terrific opportunity. Dr. Shah was appointed Maine CDC director by Governor Janet Mills in June of 2019, probably not anticipating a global pandemic to occur only a few months later. He came to Maine with broad experience in public health, most recently as director of the Illinois Department of Public Health, where he was key in implementing initiatives to address the state's opioid crisis, reduce maternal and infant mortality, and reduce childhood lead poisoning. He has both medical and law degrees from the University of Chicago, and he also studied economics at Oxford University. As an attorney and public health economist, Dr. Shah has advised professionals and governments around the nation and around the globe on improving the delivery of health care. Here in Maine, he's emerged as an incredibly trusted and respected leader in guiding the Maine CDC and our entire state through the COVID-19 pandemic with the wonderful folks on his teams. In particular, I'd like to thank you, Dr. Shah, for your great partnership that you have enabled between Maine CDC and the University of Maine system. We work together on a number of pandemic-related issues, particularly, of course, as we see cases on our campuses and get the advice and guidance of your organization. Over the course of the full pandemic, the state of Maine has held one of the lowest case rates in the country, provided ready access to COVID-19 testing, and now has launched a very successful vaccination effort that's open to everyone over the age of 16. Whenever I can, I watch Dr. Shah's regular briefings, and I urge you all to do so. Through his clear press briefings and public appearances, he's acquired quite a national following because he can provide really direct, clear communication without jargon and filled with memorable metaphors. He has a Facebook fan group called Fans of Dr. Nirav Shah with over 38,000 members, t-shirts and goods that feature the slogan "In Shah We Trust," and his own chocolate Shah bars produced by Wilbur Chocolate in Maine. I just cannot imagine a person whose message about the importance of science in our society right now could be more timely and more relevant than Dr. Nirav Shah. I thank you so much for being with us today, and it's my pleasure to welcome you on the behalf of the University of Maine to the UMaine Student Symposium.
Gosh, well, thank you so much, President Perini-Mundi. It is such an honor and a delight to be with you and with everyone today and all the students. I'm very much looking forward to the discussion. But let me first thank you for the generous invitation. We are all going through so much right now, and I have found that taking time to meet with and hear from college students about what's going on and to take their questions. They have those events, those opportunities have been some of the more engaging and invigorating conversations that I've had over the past year, and I know today will be the same. So thank you for the invitation to join, and I'm very much looking forward to the discussion. So without any further ado, what I would like to do is take a few minutes today at the top to set the table for where we are with COVID as a state, as a country, and then pose some questions that I would love to get your feedback and your ideas around, at least one, maybe a few. And then open it up to questions, either around the things that I've posed or anything else that's on your mind. Perhaps it's scientific, perhaps it's societal, perhaps it's psychological, perhaps it's policy related. Please shoot your hardest questions to me, particularly if you disagree with something that I've said, which takes me to what I want to talk about today. And that is the first, where we are with COVID, and then secondly, the value of disagreement. Let's start with where we are with COVID. It seems like over the past year, just when we thought we were about to turn a corner, we ran headlong into another surge or increase in cases. And that is unfortunately what's happening in the Northeast or in the East right now. Cases have been going up across the eastern part of the United States. For a number of reasons, which I will comment on, but even here in Maine, today alone, we logged 572 new cases, and the reports of 5 additional individuals who have died with COVID. So all of that remains concerning. But it comes at a time when we are continuing to move apace with vaccination. I'm particularly proud of the vaccination rates that we've achieved in Maine. Not that, Not that I'm keeping track of these things, but according to at least one tracker that's maintained by Bloomberg, we, as of a couple of days ago, are now the top state in terms of the percentage of the population that has been fully vaccinated, and are just in the number 2 slot in terms of states where we've gotten at least one shot into arms. Palau is still ahead of us, so I've got a little bit more work to do so that we can top the league tables. But hey, my hat goes off to my counterparts in Palau. But we are making great progress. But it's at a time when the virus is still transmitting, and it's transmitting at, sadly, an even higher rate. One of the messages that I want to convey to folks today is that a lot of the transmission that we are seeing right now is sadly on college campuses. We are seeing an increasing number of cases, an increasing number of outbreaks, and an increasing number of hospitalizations in younger people in Maine. So we're not out of this yet. And much of the impact that's being borne right now is less on older Mainers, because many have been vaccinated, but now on younger Mainers. So we're not out of this yet. We still have more to do. But I'm hopeful overall and that we will start turning a corner with respect to COVID because of the good architecture that we've put into place when it comes to vaccination, to say nothing of things like mask wearing and testing. But what I really want to talk about today is COVID in the broader context of a couple of things. The first is COVID as a lesson for science and science communication. At perhaps no time in recent history have the methods of science, public health, epidemiology, virology, you name it, have at no time have they been put front and center in a way that they are now. The public has received in real time a day-by-day lesson in the methods of science, public health, and communication. That's an important lesson for all of us who are involved in scientific endeavors. Much of what we do often is behind academic doors and discussed in journals, and that's fine. But now, when the public is paying so much attention, it raises questions because scientific principles, scientific work is not easily conveyed in a soundbite. But it also provides us an opportunity, both to show the public what we do, as well as to make the case for why what we do as scientists is important. Each of you has an important role to play in that. You can take time out of your days to talk about and help translate what you are seeing and hearing and doing as it relates to COVID for those that may not have that same vocabulary. That is more important now than it ever has been. The second thing that's critical right now is the very clear value of research. It's another reason why the partnership that we have with the University of Maine system is critical. Part of the nature of our response with COVID is a governmental or public health response, but part of it is a technical and research-based response. Research has never been more important than it is right now. Think about how much we have learned as a society, as a globe, about COVID-19 in the past 14 months. There was no COVID-19 14 months ago, 15 months. And now there are entire journals dedicated to it. Science is now more important than it ever has been. But it's not just science that's critical. It's the interdisciplinary nature of the work that we do. Another reason why universities are so critical right now. It's not just virologists that got us here. It's not just mathematical epidemiologists, and it's not just economists that got us here. It was the interdisciplinary work of all of those groups, sharing information at light speed. The concept of a preprint was not something that was around when I was in medical school. And the concept of sharing posts about your recent findings on Twitter before you even had time to type them up in an article is something that has never been done before, even during Zika virus, which was just a couple of years ago. So that nature, that interdisciplinary work has been critical to getting us where we are right now so quickly. But it also means that we have to learn to work together. And scientists often are fond of staying in their lane. You've all heard that expression before. You got to stay in your lane. You got to stay in your lane. There's a value in staying in your lane. But staying in your lane is predicated on this idea that we're in a pool, right? We're all swimming in our lanes. And there's value in that. Staying in your lane means you develop deep expertise. But it also means that you don't necessarily see what's going on in the other lanes. And so the metaphor that I think is a far more appropriate one for where we are right now in an emergency situation with so many people needed to get the job done is not people swimming laps in a lane and staying in those lanes, but rather a water polo team where everyone is swimming in the pool generally toward the same goal. That is, I think, a much better metaphor for how we need to be organizing ourselves as universities, as researchers, as students, and as public health officials in a pandemic. If we all stay in our lane, it's very difficult to see what else is going on. But if we're all swimming toward the same goal, that's a lot more effective. The other way we get there is through something that's very important to me, which is disagreement. Spirited, respectful dialogue and disagreement are the surest way to reduce the chances that your organization, whether it's your lab, whether it's your dorm, whether it's your college, or whether it's your agency, encouraging dissent and respectful disagreement is one of the easiest and surest insurance policies that you can take out to make sure you don't make a bad decision. It's been something that I've encouraged amongst my team so that if folks think that they see the world differently from the way I do, I encourage them to speak up and say, hey, I have a different idea. And even if it goes contrary to the orthodoxy that I've been thinking about, it means that even if I disagree with them, I've at least internalized what they think and go into the final decision that much more well-informed. This is what universities do best. Universities are places where research happens, where collaboration and interdisciplinary work are encouraged and taught, and disagreement and debate are encouraged and valued. That's why universities are so critical and why the University of Maine has been one of our finest partners throughout the pandemic. So I'm going to stop talking now, but before I do, again, feel free to ask me questions about anything, particularly challenging questions, but I also welcome you to weigh in on the following question. Throughout the past 8 months, the case numbers and rates of influenza, seasonal influenza, have cratered across the country as well as in Maine. That's not a surprise based on what we know right now. That's a direct function of wearing masks everywhere, physically distancing, avoiding large groups, closing down businesses, restricting indoor gathering limits at a number of different places, and putting into place strong measures that reduce the likelihood of transmission. And as a result of that, as a country, we've probably avoided the deaths of about 60,000 people from just the flu. The question that I have for everybody is whether we have an obligation to do that every single year going forward. Do we have an obligation from October through May, pretty much every year now, to put into place the same measures that we've had in place now? Doing so, of course, would perhaps avoid on the order of 60,000 deaths, fewer in some years, but more in others. But of course, would come at significant cost, directly, indirectly, and otherwise. The question that I think all of us have to grapple with is based on what we've learned about COVID, what will we do now to prevent subsequent waves of death in the future, whether from the flu or from other infectious diseases? It's a very difficult question. And whether we have an obligation to put those same measures in place in every subsequent year is one for which there's no easy answer. But I welcome your thoughts on it. So I am going to stop talking, and I would love to hear from all of you.
Thank you so much, Dr. Shaw. There were so many important messages that you have relayed. And of course, I'm sure that people are going to respond to your question about influenza. I'm going to start off with some of the questions that we've received thus far. One is, you mentioned that there's a growing number of cases in the state of Maine. Is there any way to tell how many of the positive COVID cases were among people who have been vaccinated?
Yeah, we keep track of positive COVID cases that among individuals who have received and completed their vaccination series. They are what's called breakthrough cases. And thus far, as of yesterday, there had been 93 breakthrough cases in Maine. To put that in perspective, I ran the percentage yesterday. To put that number in perspective, it's approximately 99.998% of people in Maine who have been vaccinated have not gotten COVID. So there is a small number of people out of the nearly 1 million shots that we've administered. There's a small, vanishingly small percentage who have developed COVID after a subsequent exposure. That's neither surprising nor shocking. No vaccine has a 100% effectiveness rate. What we're trying to do is make sure we're tracking those cases so that we can see what's going on. Now, here's what's also very interesting. Of those 93 cases, all of them had extremely mild symptoms. And I forget the exact number. I think 2/3 of them had no symptoms at all. So even at the point that they got COVID, the nature of what they got as COVID was exceedingly mild. And that is further proof positive that the vaccine works.
Great, thank you. Someone has asked if there's any new information about long-term immunity with the vaccines, and if you could comment on that.
Well, long-term immunity is interesting when you think about vaccines, because in order to gauge long-term immunity, you have to have long-term data. And the vaccines that we have right now were studied in clinical trials that started round about 10 months ago. And we are now just doing the second waves of follow-up. And so it will be some time before we have enough data to really gauge what the degree of long-term immunity is. But based on the data that we have right now, the immunity with the 2 mRNA vaccines, which have been on the market and studied the longest, it lasts at least for 6 months, but every single virologist and immunologist with whom I've spoken is almost assured that it will last for a longer period. Now, how much longer is not yet really clear. Yesterday, some scientists and the CEO of Pfizer indicated that some individuals may need to get a booster shot in round about a year. It's too early to tell. Some of that is a function of how much immunity is stimulated by the vaccines. Some of it's also a function of how the virus changes. We shouldn't be, we shouldn't be overly focused on that. We should be focused on it, but keep in mind that if you've ever gotten the flu shot, then you've experienced a situation where a virus has changed, necessitating a new vaccine every year. If that happens with COVID, so be it. And I've heard from scientists at Moderna and Pfizer who have indicated that they are already planning and preparing for how they may adjust the vaccine or create booster shots if needed.
Okay, I'm going to combine a couple of questions here. So one is that Maine has had such a successful vaccination campaign. So what are the factors that have led to the state's success in that campaign? And kind of on the flip side of that is that now with the new data that has been released about the potential correlation with the Johnson & Johnson vaccine and the blood clots, this might lead to some vaccine hesitancy. So how can we deal with vaccine hesitancy in our society, and how can we communicate effectively with people who are feeling hesitant?
Great. So let's first talk about where we are in Maine. I'm so incredibly proud of the job our state has done with respect to everything involving COVID. The success we're having in vaccination should be no surprise to any careful observer who's seen what we've done previously. We have achieved one of the lowest rates of infection of any state. We've achieved the lowest rate of hospitalization of any state, notwithstanding our older than average population. And now we have achieved, not that I keep track of these things, but one of the top rates of vaccination, if not the top rate around. Again, I give credit to my colleagues in Palau, but we're coming for you. We're gonna try to surpass them as well. I think there are a few things that are going on there. The first is just purely denominator driven or numerator driven. That is to say, in order to achieve high vaccination rates, you have to have a state of residents who want to get vaccinated. And in a number of states right now, they are hitting hesitancy walls, Alaska, Mississippi, et cetera. They are finding that they have topped out around the number of folks that wish to get vaccinated right now. We haven't seen that in Maine. Actually, just this morning, I was down in Portland at the Portland Expo, where there's a large scale vaccination site, and they put appointments up and they get snapped up immediately. So we're still seeing a lot of urgency to get vaccinated. That's one reason I think we're doing well overall. The second, though, is the partnership that we've struck in Maine, by and among various different groups of vaccinators, I think sets us apart. We've got a working group comprising not just Maine CDC folks, but hospitals, EMS clinicians, independent pharmacists, FQHCs, all of whom have come together to say, we're going to make this a priority and we're going to put other things on hold to get it done. That's not been the case in other states. And I think that's another reason. The third is, I think some credit here goes to the system that we put into place. It was heavily criticized, to be sure, but I'm referring to the approach that we've taken, which is just to go by age. The rationale behind that was that it would be the quickest. And in the midst of a pandemic, where 5,000 people are dying every week in the United States, the quicker we vaccinate, the quicker we keep people safe. And I do believe that approach sped up the process, and that's helped us get to where we are.
There's one question that is more of a personal nature here. So you have been in the spotlight since the beginning of the pandemic, which was really at the start of your position at the Maine CDC. So how have you handled being in the spotlight for all this time?
Well, I appreciate that question, and it's interesting and ironic in a few ways, because one is that it's not my desire or intention to be in the spotlight. In fact, almost all the advice that I give people is so that COVID goes away and I no longer have to be in the spotlight. But there are a few important things. I mean, I think what has to be said right up at the top is to recognize that spotlight notwithstanding, my family and I have been able to contend with COVID in a way that a lot of Maine people haven't, right? I've got a stable job, we have a safe place to live, and other people in Maine have been a lot more affected by COVID, and we've been privileged to be relatively safe in that regard. But it is not, that doesn't come without its challenges. And you're right, being in the spotlight in and of itself raises the risk level of every decision I make. You know, the way that I handle it is twofold. The first is never sugarcoat anything. In fact, I have a rule when I speak with anybody, which is that if someone asks you what time it is, do not tell them how to build a clock. When someone asks you what time it is, you should tell them what time it is, even if it's not the time that they were wanting to hear. And I think that's one reason that I've been able to not just be in the spotlight, but not necessarily attract the slings and arrows that counterparts of mine in other states have sadly experienced. The other principle is, you know, COVID's tough. It's dark. It's not just metaphorically, but literally dark days. And even though the work that surrounds COVID, it can be morose, I try not to be a doomsayer. There's bad news out there. There's always bad news. But I'm not shy about sharing good news. And sometimes I get criticized for that. I get criticized for minimizing things. But I believe that humans can feel simultaneously scared and sad about bad things, and also happy and optimistic about good things that are happening. And that's why I'm not shy about tossing in the occasional dad joke from time to time, because the work itself is dark enough. That doesn't mean we have to let it color our worldview.
So going forward, how do you think this might change our life in a more permanent kind of way? And you had mentioned that we've been able to squash influenza this season. And so going forward, what are your thoughts on wearing face coverings, physical distancing, and maybe also the requirement for vaccines for COVID-19 and for influenza? If you could comment on kind of all of that, because we have so many questions here.
Okay, so let's take each of those in turn. There are the larger scale societal questions about how we plan and prepare for the next pandemic. I'll get to that in a minute. But more tactically right now, the immediate questions facing us are how we think about vaccines. And there are two axes of that question. The first is what you referenced, which is whether any vaccine should be mandated, whether at the workplace site, at the college or university level, or elsewhere. That's a question that's going to take a little bit of time to sort out. One of the reasons is that there are still some regulatory hurdles that are in place. I also don't know whether as a society we really zeroed in or honed in on what we think the nature of a mandate should be versus incentives to just be vaccinated. The other question, of course, that's coming at us for the summer are these so-called vaccine verification systems and what we think about those. So those are big questions. The other set of questions about the way things will change has to do with how we think about big events. So pandemics fall in an interesting category of events. They are in the category of high magnitude, low likelihood events. Things that happen very infrequently, but when they do, cause bedlam. That's contrasted with high likelihood, low magnitude events. Things like an outbreak of diarrhea, food poisoning at a church picnic. The way we think about these high magnitude, low frequency events, it has not been right for the most part. Let me. I was talking to a friend of mine recently about this, who's a writer. Some may be too young to remember this, but many years ago, there was a national scare with respect to individuals who had ingested Tylenol that had been tainted by cyanide. Many of you will remember that. I was talking with a friend of mine, about a month ago, who was a writer who writes about medical type issues. It was a fascinating conversation. And we were remarking on the fact that after that event happened, a very low likelihood, but high magnitude event, society pretty much decided that that was never going to happen again. Everything changed. Governmentally, manufacturing wise, the maker of Tylenol, a company you may have heard of, Johnson & Johnson, decided to change everything about the way that the drug was made, manufactured, packaged, and presented. We decided as a society that was never going to happen again. So what are we going to do with pandemics, right? Are we going to say, you know what, this is too much, we are not going to let this happen again? Or are we going to treat pandemics the way that we do car crashes, which is to say, car crashes happen. It's an unfortunate fact of life. That's how it's going to be. That's the big question I think that's facing us.
Well, I think we only have 1 minute left with you. And I just want to ask one final question for the students. So for those students that are interested in a career in medicine or public health or science policy, can you provide some career advice for them and maybe some advice about how to be an effective communicator?
Good. So let's talk about the latter first, because being an effective communicator is really critical, because the work that you do has implications for your field, no doubt. But the way that we communicate about those is with our peers in journals and at conferences. But nowadays, the work that you do also has implications for the broader context, and being able to convey that is critical. But these skills can be taught, they can be learned. You can practice them yourselves. You know how I test messages? There's one person in all of our lives who will always pick up the phone when we call. And that's our mother. And what I do before I am about to deliver a difficult or complicated or hard message is I call my mom the night before. And if I can't, I used to teach my debate students this when I would coach debate. If you can't explain a concept in about 30 seconds to a minute to your mom, then you're not going to be able to communicate it to the broader public. And so there are ways to do this and practice it. And I encourage everyone to do so. It's a skill. And like all skills, you can learn it and you can get better at it. Now, in terms of those who are looking forward toward careers in science, medicine, and public health, I absolutely encourage you to do that. And the pandemic has shown the world what we need and what we can do. My only piece of advice there, it's meta advice. My only piece of advice is to be skeptical of advice. And that is because in my own career, I would go to folks and seek out their advice. And invariably, they would tell me to do what they did. Confirmation bias of a sort. And eventually I kind of realized, gosh, why am I asking people for their advice? Why don't I just do what I'm interested in? And maybe that'll work out. Maybe it'll be a spectacular failure, but I will just go in the direction that I was interested in. And that is why I opted to go to law school rather than finishing my economics PhD, because I was more interested in regulation rather than the mathematical side of economics. And that's why I went to law school, and that's why I worked in the private sector, because that's where the issues were interesting. And so I ask everyone to seek advice from mentors, but also look for people who won't just tell you what they did. Look for mentors and people that actually do something crazy, which is to ask you what you want to do and then help you get there. Those are the folks who will really make a difference in your career. There were two in my career. There were two folks who didn't just tell me to do what they did, but rather said, Nirav, what do you really want to do? And they are in no small measure the reason that I'm chatting with you all today.
Well, Dr. Shah, thank you so much for joining us today on behalf of the University of Maine and all of our attendees today. And thank you for your tireless efforts to keep Maine safe during the COVID-19 pandemic. Thank you so much.
Thank you so much all for this opportunity. Real pleasure to chat with everybody.
Absolutely. The following presentations highlight a sample of the amazing research projects and contributions from our undergraduates and graduate students in a range of technical categories. The students will introduce themselves and provide an elevator pitch of their research.
I'm Anna Schumann. I'm a senior at the University of Maine, and I study molecular and cellular biology. I'm part of the Molar Lab, where we're interested in antibiotic resistance of Mycobacterium abscessus. As panel 1 shows, Mycobacterium abscessus is extensively resistant to antibiotics, and treatment is only successful in 30% of all cases. Interestingly, when a group of clinical researchers isolated antibiotic-resistant clinical isolates, they found that almost all isolates carried at least one prophage, which are viruses that infect bacteria and integrate their genome into their host genome. While it is known that prophage contribute to antibiotic resistance in some bacteria, this relationship has not been investigated in mycobacteria yet. In our studies, we used model organism Mycobacterium cheloni, and we were able to show that the presence of prophage McProf changes the bacterium's response to stress, as shown in panel 2. You can see that when McProf was not present, there was only a small response to stress. But when McProf was present, this response was significantly increased, as seen in an increase in antibiotic resistance gene expression and antibiotic resistance. We wanted to know what genes of McProf could potentially cause this difference. So we looked at its genome, as shown in panel 3. The red and blue uptakes show which genes are turned on in the bacterium and might be able to contribute to the bacterium's response to antibiotics. While McProf did not express any genes that directly confer antibiotic resistance, it expresses a polymorphic toxin system. These genes are present and used by many bacteria to sense stress and communicate to other cells in their population. So we think that McProf might use this system to communicate to its host. While we're not entirely sure how it works, based on our research, we think that these genes are secreted, either directly into the medium or delivered into the other cells in the population. We think that they are triggered by stress and then cause an increase in antibiotic resistance gene expression and antibiotic resistance in the overall population. We aren't entirely sure if it happens in the cell that's triggered by stress or in other cells. But by expressing the polymorphic toxin system itself without any of the other prophage genes in our model organism, in situations of stress, I was able to show that some antibiotic resistance genes are increased. I yet have to show whether the antibiotic resistance is also upregulated. But my preliminary data show that the polymorphic toxin system might be important for antibiotic resistance. And if that is confirmed by later data, we might be able to learn how to target these genes and prevent antibiotic resistance from developing. Thank you.
Hello, everyone. My name is Emily Roth. I'm a fourth-year undergrad in the School of Forest Resources and Women's Gender and Sexuality Studies. My research mentor is Dr. Jessica Leahy. Forestry professionally and in universities is dominated by men, and university forestry programs struggle to bring gender minorities into programs and to maintain their presence. And gender minorities in this context refers to women, non-binary people, and other non-men genders. Gender exclusion is perpetuated in one way through gendered language, which is, in this context, biased language favoring men, like using, for example, he, him pronouns to broadly refer to an all gender group. Past research has explored the lack of diversity in natural resources and suggested the importance of gender fair language for inclusion. But to date, there has yet to be a study that considers the intersection of gendered language and undergraduate forestry student experiences. The big picture objective for this research is looking at how undergraduate forestry student experiences in their program are affected by gender, with an emphasis on gendered language. And these are the findings that I'll focus on in this presentation. I took a qualitative case study approach for this research, and I coded thematically. So I interviewed gender minorities and majorities from two major forestry land grant universities in the US. And I asked questions like, what have you noticed in terms of exclusion within the forestry profession? And can you share examples of gender inclusion and gender exclusion you have seen in the classroom? And then I coded these 24 interviews using Taget software. So preliminary findings indicate that the exclusion of gender minorities in forestry is perpetuated by the use of gendered language, and the use of gendered language is common. And the gender minority from both universities were generally on the same page that gendered language makes forestry feel inaccessible and unwelcoming, while gender fair language promotes feelings of belonging and opportunity in forestry. Gender majority opinions, however, vary. Most participants claim to not notice or feel unaffected by gendered and gender fair language. And two students mentioned feeling that forestry was uncomfortably hypermasculine and unhealthily competitive. Alternatively, one student claimed that feminism is too pervasive in forestry, and asking about pronouns makes them uncomfortable. So in section A on this poster, I made a word cloud of the interview transcripts. In section B, I noted a few powerful quotes from the interviews, and I won't read them all, but one that was particularly powerful for me was from a gender minority student, and they said, when men professors refer to future careers, it's usually geared more towards men, and it just feels like there's less opportunities for women, or you have to try a lot harder to get some of those same jobs. And section C is a graph that I created from a Qualtrics volunteer form that I used to screen interview candidates. I asked whether students had noticed gendered language in their program, and you can see that the gender majority's answers varied, but the gender minority predominantly answered that yes, they had noticed gendered language in their program. In short, based on the research objectives, preliminary results indicate that gendered language negatively impacts the gender minority by making forestry feel inaccessible and unwelcoming. But a positive note that came from this research is that many students cited increased use of gender fair language in the classroom since they came to college. And I invite you to read my full poster for more details. And thank you so much for the opportunity.
Hi, my name is Christine Hale. I'm a fourth-year PhD student in the Graduate School of Biomedical Sciences and Engineering. I carry out my thesis research at the University of New England in Dr. Jeffrey Ganter's lab, which is within the Center of Biomedical Research Excellence in Pain and Sensory Function. It's estimated that 50 million adults nationwide are affected by chronic pain. And whereas we do have effective treatment options for acute pain, such as opioids, we now know that these drugs do come with some severe side effects, such as addiction, which has led to the opioid addiction crisis that our country is currently facing. So to combat this crisis with the discovery of new drugs, our lab investigates the genetic mechanisms of nociceptor sensitivity, which is involved in the pain-signaling pathway. And we do this using the fruit fly, which shares 75% of the genes connected to disease in humans. Using this model, our lab has already identified 13 novel genetic components within the BMP signaling pathway as being involved in nociceptive sensitization. And so we know that our approach is effective, but we still have some important gaps left to fill, such as, are there any other biochemical signaling pathways involved in nociceptor sensitivity? So to answer this, we recently focused on a gene within the Wnt signaling pathway named armadillo, or ARM for short. It's also known as beta-catenin in humans. The methodology of our research includes a genetic manipulation and behavioral assessment approach. So we can investigate genes that we hypothesize may be possibly involved in nociceptor sensitivity, such as armadillo, by either knocking down or enhancing that gene's expression within the nociceptors. And since nociceptors are sensory neurons, if the gene is necessary for their sensitivity, then we can visualize this with thermal behavioral tests that we carry out on our animals, which measures their pain level. And so following this methodology and investigating armadillo, our findings showed that when armadillo was knocked down in its expression within the nociceptors, that animals showed significantly less pain. When armadillo was overexpressed within the nociceptors, our findings showed that these animals showed significantly more pain. A thorough analysis of the dendritic structure of these genetically manipulated neurons showed no significant change when compared to controls. So based on these findings, we were able to conclude that armadillo does play a role in facilitating baseline nociceptor sensitivity, and this is not by morphological changes, but possibly by signaling mechanisms. Also, the discovery of armadillo's involvement in nociceptor sensitivity potentially opens up other genes within the Wnt signaling pathway that could be investigated for drug target testing while using our fly model. And really, this just gets us one step closer to our ultimate goal, which is to combat the opioid crisis, even here in Maine, by discovering potential new drug targets for treating pain. Thank you.
And next up, we have Lee Parrish. Lee, are you with us?
I can see her talking and it says she's unmuted, but
Hello?
there's no sound.
Lee, are you there?
I am. Hello?
Okay, great.
You can hear me?
I can hear you now.
Oh, I'm so sorry. Thank you so much. Okay, hello, and thank you for having me here. My name is Leah Parrish. I'm an undergraduate here studying computer science with a minor in political science. I work at the Multisensory Interactive Media Lab with Dr. Namesha. Over the course of the academic year, we have performed a completely remote study investigating the effects of digital games and essential oils on work stress recovery called the exploration of olfactory interventions as a synergistic emotional and cognitive recovery technique. Work stress is, of course, a huge issue and has only become more significant throughout the COVID-19 pandemic. We know that stress has extreme negative physical health consequences, including, but not limited to, increased blood pressure and heart rate. The American Psychological Association has an article called Stress Effects on the Body, published November 1st of 2018, which outlines that stress affects virtually every part of our body, from muscle tension, putting us at risk of pain and injury, to the ability for acute stress to cause asthma attacks and otherwise, quote, exacerbate breathing problems for people with pre-existing respiratory diseases. Clearly, stress is a public health concern. So in approaching stress incurred through remote learning and Zoom fatigue, we designed solutions specifically targeting students who were working from home as an attempt to encourage psychological detachment from work. I designed a small sliding box in SolidWorks you can see on my slide. And the study consisted of people going through work and relaxation tasks while tracking their productivity and emotional states, looking to see how these work and relaxation tasks affected recovery through analyzing the recovery outcomes available to us. The general path of work and relaxation was the same for every person, but the recovery technique was changed for each person. The control group actually had no recovery and would only work. The second group was game only, meaning they worked, played a game for 10 minutes, and worked again. The third group was game and sent, and in this group, we would mail each participant one of the sliding boxes. Then the participants would work, open the box and play a game for 10 minutes, then close the box and work again. The work section consisted of doing single-digit multiplication problems for 10 minutes. We do not have any definitive data to show yet, and we are continuing our study over the summer, along with developing additional technologies to complement and expand on our research, such as a scent clock that will signal events through non-visual, non-auditory means. Our study has impacts on people's well-being and long-term health, as reducing stress is imperative, and we focus on identifying recovery techniques that are the most effective catalysts of this stress reduction. It has impacts on industry, as opportunities for technological or economic developments are unveiled through the lens of our research, and as new methods for increasing productivity and employee engagement. It has impacts on how we interact as a society with our world and computers, and how we choose to take care of ourselves. Our study will contribute data to the growing base surrounding the benefits of human-computer interaction, human psychology, and the growing investigations into olfactory augmentation in diverse settings. I also believe that our methods could be a start to doing more controlled remote studies and using the internet for more intensive research endeavors. Computers are outstanding tools, but they can cause a plethora of issues that perhaps we can use the same tools to solve. Prioritizing the well-being of every member of society is to prioritize the well-being of society. Again, thank you for having me here. I'm Leah Parrish of the MIM Lab. Have a great day.
My name is Amy Frankel. I'm a graduate student in social work. I am completing a research study with Sydney Massa and Morena Slover, who are also graduate students in social work, and our faculty sponsor is Elizabeth DePoy. We are conducting a study about the stigmatization of formerly incarcerated citizens. More than 600,000 people are released from state and federal prisons annually, with nearly 95% returning to their immediate communities. Despite the surge of interest and empirical study of this topic, the literature investigating prisoner reintegration has largely neglected the subjective perspective of formerly incarcerated citizens about what it means to live with the ex-convict label. The aim of this study is to begin to fill the knowledge gap, to examine the nature of stigma in the lives of individuals who share the experience of having been incarcerated and who have spent varying lengths of time back in their communities following their completion of a term of incarceration. We aim to provide greater understanding of the impact of a history of incarceration on returning citizens and expand research to capture the subjective experiences of formerly incarcerated citizens in order to identify stigma and inform strategies for social change and opportunity. Our research questions are: what is the degree of stigma experienced by formerly incarcerated citizens? And to what extent does this population offer insights about sources and consequences of stigma beyond what is found in existing literature? Using purposive sampling, we are conducting phone interviews consisting of closed and open-ended questions, and then using statistical and thematic analysis to analyze our interview data. Participants for this study are formerly incarcerated citizens, 18 years of age or older, who are not currently on probation. Preliminary findings from our interviews reveal insights that we anticipate will be articulated and enriched further. We see themes emerging around the amplified complexity of family relationships while incarcerated and after release as well. Similar issues manifest in navigating interactions with non-family members in the communities of returning citizens. We are hearing testimony about respondents opting to change their work and home locations in order to avoid the feelings of stigma they experience in their original communities. One notable data point is the ongoing feeling of stigma, even years after release and regardless of the accomplishments of the respondent. We are still in the interview and data gathering process. As the study proceeds, it promises the following knowledge outcomes. It is likely that the experience of stigma truncates re-entry in all spheres of life. Lack of opportunity resulting from this stigma increases the chance of recidivism. It is important and powerful for participants to have a voice in resolving their own social problems, an opportunity and direction for the next study in this important research agenda. Thank you.
Hi, everyone. I'm David Flewelling. I'm an undergraduate biomedical engineer, and this presentation is done in coordination with the Mount Desert Island Biolabs and the INBRI program on Pseudomonas aeruginosa infection interacting with the NF-κB pathway and integrin signaling. Pseudomonas aeruginosa is an opportunistic pathogen. It usually does not impact people, but it can be common in those who have compromised immune systems or other forms like maybe a disease or a post-operative immune weakness, like a interaction with immunosuppressants, which can lead to a rooted infection in the person, and it can be quite severe, significant impacts on mortality. It's sometimes the cause of death in patients. And the ways that this interacts with the cells are not fully known. So we theorize that you can examine the way that it interacts with cells through the genetic pathway. So to measure this, we used a model animal, the zebrafish, which is used very often at the university because it has such a rapid cycle of development and some similarity to human gene expression. And we added a cystic fibrosis morpholina, which is a common condition that leads to Pseudomonas aeruginosa infection, as well as exposure to lead, and then measured the relative expression of different gene segments of the zebrafish embryos using RNA sequencing. And then with this, we evaluated different levels of transcription within the embryos to determine which genes were upregulated. And we found that there was a dramatic upregulation in several genes, but notably the ITGB3 alpha gene, which codes for part of the integrin pathway, which is a key part of the immune system. So further investigation is still needed. This is just an initial analysis of the data. We don't fully know the panel of genes that are affected and the aspects of the integrin pathway. But this does confirm what we suspected, which is there is an interaction between Pseudomonas aeruginosa and gene transcription in different parts of the cell immune response.
All right, so hello everyone. My name is Michaela Moore, and I'm a first-year PhD student in the Graduate School of Biomedical Science and Engineering. And with this program, I was given a great opportunity to work on a rotation project in Dr. Vivek Kumar's lab of the Jackson Laboratory located in Bar Harbor, Maine. So everyone gets bored at work or at school, and who knows, some of you may even be bored right now, and that's okay. But for some people, staying focused on a given task can be very difficult. And this difficulty doesn't just arise out of boredom or perceived lack of motivation. It arises from an underlying neurological condition that is known as attention deficit hyperactivity disorder, or ADHD. For those of you who may not know, ADHD is the most commonly diagnosed psychiatric disorder among children in the United States, affecting nearly 6 million individuals, where this number continues to grow each year. We also know from published reports that ADHD can not only have a severe negative impact on academic and social functioning in children, but this disorder most often persists throughout adolescence and into adulthood. Despite the high prevalence of this disease, the etiology of ADHD still remains unknown. As researchers and scientists who look to improve the quality of life in individuals, we often turn to animal models to help us better understand the biological changes involved in particular behavioral diseases. To date, no sufficient mouse model exists for ADHD, so my lab and I developed a mouse model that recapitulates the disease symptoms to help us better understand the molecular mechanisms involved. Our model, known as the Crichton model, develops increased hyperactivity in an age-dependent manner, allowing us to elucidate the molecular changes in both children and adults. With this new model, we are able to take a closer look into the ADHD brain. Using imaging and antibody analysis, our data shows distinct neuronal changes occurring in regions of the brain, such as the prefrontal cortex. Further, we as researchers like to look deeper. We know from the literature that pharmaceuticals are the most common course of treatment for individuals diagnosed with ADHD. However, many aspects on how certain drugs can impact the developing brain are still unknown. With this model in our lab, we assessed on the transcriptional level what changes are occurring that lead to ADHD symptoms. Not only is this important to help us better understand how biology and chemistry can impact our behavior, but it can also help us find and develop new drug targets to better treat ADHD. I'd like to give a sincere thank you to my mentor, Dr. Vivek Kumar, my colleague Yaya Barakrat from Tufts University, and my colleague Arojit Mitra for their guidance and input on this project. If you have any questions, please feel free to email me at michaelamore@umaine.edu. Thank you.
Hello, my name is Bailey Woodard, and I'm a fourth-year undergraduate bio-pre-med major at UMaine. I perform my research in Christy Townsend's lab, which is now located at The Ohio State University. This research focused on the development of a pressure myography system to test the vascular integrity of aged vessels. Our lab has previously demonstrated that pathologies such as aging and diabetes can result in a dieback of nerves, also known as neuropathy. This disorder also reduces neurovascular interactions in peripheral tissues. As the nerves around these vessels begin to dissociate, signals that would usually be sent to the cardiovascular system are lost. This reduces the ability of aged blood vessels to respond to stimuli as well as maintain a healthy flow rate. In order to investigate this phenomenon, we sought to find a technique that measured the vascular function of small resistance vessels in the adipose tissue of aged mice. It's also important to note that the vessel that we chose to study has a width of about 4 human hairs, so this limited our options until we stumbled upon the field of pressure myography. You're probably wondering by now, what is pressure myography? Well, this technique first involves dissecting a blood vessel before removing all the surrounding cells so that the vessel wall can be visualized. After this, the vessel is cannulated at both ends and placed into a vessel chamber. While in the chamber, the vessel is surrounded by a physiological solution, which is both pressurized and heated to a degree that the vessel can be considered at a physiological state. When chemicals are added to the solution, the vessel will contract or dilate, and the amount of vascular relaxation and vasoconstriction is measured in real time using a USB camera. After doing a bit of research, we soon realized that the pre-made system would cost tens of thousands of dollars. It was at this point that I became interested in building my own pressure myograph, even though I had no prior engineering experience. I soon learned about a company called VasoTracker that has an online repository including materials lists, instructions, and software downloads that could be used to build a pressure myograph at a fraction of the cost of commercial alternatives. As you can see in the second image, this is a very complex setup involving many components in order to keep the vessel at a near physiological state. In order to get the myograph to work, the vessel chamber and pressure monitor had to be assembled by hand. It took me around a year to build and set up the system, and I did all of this virtually independently as my lab moved across the country last summer. After a lot of troubleshooting, we were finally at the point where we can start collecting data using the homemade pressure myograph. In fact, I'll actually be doing my first experiment today. There are multiple uses for the system, as it can be used to test the functionality of vessels throughout the body under different conditions, and it can also be used to help find cardiovascular treatments for humans as well. This is especially pertinent in our lives right now, as the median age of Maine's population is steadily rising. and the number of diabetes diagnoses are climbing across all demographics. I'm excited to contribute to this rapidly developing field, and I'm confident that future data collected on this MyGraph system will make a difference in the lives of many individuals to come. Thank you.
My name is Brandy Seuss. I'm a third-year graduate student in molecular and biomedical sciences. I conduct my research in the lab of Dr. Ben King at the University of Maine. Our lab studies the innate immune response to pathogens, where I specifically focus on the neutrophil response to influenza type A virus. Now, for those of you who don't know, neutrophils are the body's first responders. They basically eat pathogens and secrete ROS. ROS, or radical oxygen species, such as hydrogen peroxide, attack invading pathogens and recruit more neutrophils to the site of an infection. Now, this can be a double-edged sword because if you're not careful, you wind up with hyperinflammation due to too much ROS produced by too many neutrophils and too much recruitment at the site of infection. And this can lead to tissue damage and subsequent death. So what we wanted to know is what causes this switch genetically and whether or not we can figure out the time at which these neutrophils switch from being helpful to harmful. To do that, we actually generate a zebrafish infectious model by injecting wild type influenza into these fish and looking at genetic factors, their survival output, how much virus burden they carry, and other factors. And what we found is that if we start manipulating certain aspects of the zebrafish involved in neutrophil regulation and ROS output, we see very different outcomes. If you look at the graph with the black line, that is our wild type line, which is our AB. This is kind of the standard that we use for everything because all of the different lines that you see labeled are actually based on the AB line. And with our wild type line, we have a death rate of about 45 to 50% stabilized, depending neither on virus, but only the type of fish that we get that day. And when we start genetically manipulating certain genes, such as in the WIM fish, when we start manipulating this pathway that actually controls the number of neutrophils that can be released into the system, we actually start seeing very different responses. With our WIM syndrome that represents a human WIM syndrome, we get less neutrophils that are both less effective and also smaller in size. And as you can see, correspondingly, the death rate actually increases by 20%. However, when we start targeting ROS producers, we get a positive reaction. When we look at spotless fish, which target a specific gene on neutrophil that produces ROS, we actually see a modest increase of about 10% survival. But when we target NCF1, which is part of a complex that produces a lot of exogenous ROS, we actually see a significant increase in survival of about 22%. So what this tells us is that NCF1 is likely involved in hyperinflammation and that we probably shouldn't mess with the number of neutrophils because our fish are going to die. However, the other thing you can see from this graph that isn't quite mentioned is if you look at the days post-infection between 2 to 4, around 3 days, you start seeing a significant number of deaths. And what I found in our studies is that between 2 and 3 days, if you haven't pharmaceutically rescued the fish in 48 hours or genetically modified them, you can't rescue hyperinflammation. Likely what this tells us is this genetic switch occurs within the first 48 hours. So what we're doing to understand it a bit more is looking at RNA-seq, as well as a lot of different time points every hour for the first 48 hours to see what genes have been changed during this. And hopefully we'll have a better therapeutic outcome and see if we can't use this to treat hyperinflammatory diseases as well as patients who suffer from viral hyperinflammation. Thank you very much.
My name is Emma Dillard, and I'm an undergraduate student in the School of Marine Sciences here at the University of Maine. My research is a part of the Marine Biodiversity Observation Network and was advised by Drs. Lee Karp-Boss and Jeffrey Runge. The primary objective of my research is to describe variability in zooplankton biomass in the Gulf of Maine over space and time. Zooplankton are mostly small organisms and larval stages of larger animals that drift in the ocean. They feed on the primary producers, that is the phytoplankton, and in turn represent the food source for larger animals. In the Gulf of Maine, Calanus fimarchicus makes up the majority of the zooplankton community. Due to its high energy stores, this lipid-rich copepod, which is pictured here, is the preferred prey species for many commercially important planktivorous pelagic forage fish and the critically endangered North Atlantic right whale. In recent decades, the Gulf of Maine has experienced a rate of warming, which is among the highest across the globe. Drastic changes in circulation of water masses in the Gulf of Maine has altered the physical properties of the water on multiple levels. Due to the important link they represent between higher and lower trophic levels in the marine environment, understanding changes in zooplankton populations will allow us to generate a better understanding of what biological implications these changes might have had and might have in the future. In this study, I analyzed existing biomass data that were collected between 2005 and 2017 at 3 stations in the Gulf of Maine, looking specifically at trends of change for each season. Biomass represents the quantity of zooplankton by weight and that is present in a water sample and allows us to estimate how much food is available to the next trophic level. One of the stations, DMC1, is located within the Damariscotta estuary. Station DMC2 is located in the Maine coastal current, which is considered important for the transfer and distribution of zooplankton throughout the Gulf of Maine. The third station, WB7, is located over the deep Wilkinson Basin, an important repository for Calanus fimarchicus in the Gulf of Maine and a historic feeding ground for the North Atlantic right whale in late spring and early summer. The plots on my slide show no clear trends of change at the station within the estuary. They show a decrease in biomass in the winter and fall and an increase in biomass in the summer in the Maine coastal current, as well as over Wilkinson Basin. These changes are more pronounced over Wilkinson Basin than they are in the Maine coastal current. Future work will focus on examining the composition of species that make up the total biomass of zooplankton in the Gulf of Maine and how this relates to biological and physical changes that are happening in the Gulf of Maine. Thank you.
Hello everyone, my name is Neeta James and I'm a third-year PhD student from the Multisensory Interactive Media Lab at UMaine. Do virtual flavors influence liking and emotions? Think about how you would feel if you have to have black coffee every day for the rest of your life. No cream, no sugar, no latte, no macchiato. Boring, isn't it? On the contrary, think about how it would be if you can have black coffee one moment and have latte another moment, but still keeping it on the healthy side. Exciting, isn't it? So why flavors? In our everyday life, we look for exciting taste, smells, and flavors in everything we eat and drink. Flavors are not just influenced by taste and smell, but also by the color, the sound, the texture, and the temperature of the food as well. It is multimodal. Traditionally, to change flavors, people have always physically or chemically altered the food. With coffee, for example, they added milk or sugar or totally converted the form of it into a latte or a frappuccino. With that, there has always been some health issue attached to it. So our thoughts were geared towards how we can change the flavors based on the changing moods and emotions of people, but still keeping it healthy. This is where virtual flavors come into play. Virtual flavor stimulates taste, smell, color, temperature externally through the utensil we are eating with without changing the food. There has been a lot of research done to understand the liking and emotions and how they change with flavors with real food and beverages. But has it been applied to virtual flavors? No, not yet. To fill that research gap, we aim to introduce a novel approach to study how virtual flavors changes flavor perceptions, liking, and emotions of people. For this study, we had 30 participants who evaluated the virtual coffee mug as shown in the picture, wherein they would be drinking just water, but with the smell and temperature stimuli, they would perceive it as different flavors of coffee. So they were asked to taste a few samples, answer a few questions to evaluate their liking and emotions, and they were good to go. We found interesting results in how emotion changes as shown in the map. There was a lot of wide range of elicited emotions, which signifies that virtual flavors do influence various emotions for people. For example, mint flavor was related to adventurous emotion. Mint plus hot temperature was related to good-natured and understanding emotion. Mint plus cold temperature was related to peaceful emotion. This represents a good user experience, which signifies that it can be ultimately incorporated into our everyday lives with eating and drinking. The results from this study open up a lot of scope for our future work, which encompasses fine-tuning the stimuli configurations for virtual flavors, changing different base drinks, and exploring more on the emotional responses with the virtual flavors. This eventually is envisioned to be built into a framework that would be helpful for the food industry, and not just the food industry, but nutrition and medical industry as well. With that, I would like to conclude my talk with a question for you all. Are you all ready for a world of virtual flavors towards a healthy and enjoyable eating experience? Thank you so much for your patience and listening.
Hi, everyone. My name is Avery Bond, and I'm a second-year PhD student in the Department of Molecular and Biomedical Sciences at UMaine. I'm conducting my thesis research in Dr. Melissa McGinnis's laboratory, where we study virus-host interactions to identify how viruses cause infection. So as we've all seen over the past year or so, viruses can wreak havoc on our bodies and have major impacts on every aspect of our lives. The COVID-19 pandemic has stolen the lives of so many, and unfortunately, it is not over yet. So in order to halt pandemics and viral infections as a whole, we need to identify treatments, whether that be through vaccines or other therapeutics. So one virus that is of particular interest to our lab is JC polyomavirus. JC polyomavirus infects upwards of 80% of the global population, so that means about 4 out of 5 of us listening right now are infected by JC polyomavirus. So fortunately for most, JC polyomavirus infection is an asymptomatic infection of the kidneys, and you might go your whole life without knowing you're infected by it. Unfortunately, for those with severely compromised immune systems, JC polyomavirus infection is able to spread from the kidneys to the brain, where it causes a disease known as progressive multifocal leukoencephalopathy, also known as PML. So PML results in the formation of lesions in the brain caused by JC polyomavirus killing our brain cells. Common symptoms of PML include loss of coordination, loss of vision, cognitive disorders, and trouble speaking, and this deterioration usually occurs rapidly. So although JC polyomavirus was discovered about 50 years ago, we still do not have any effective treatments. So in an attempt to identify treatments for JC polyomavirus infection and PML, our lab conducted a wide-scale drug screen where we received hundreds of drugs and inhibitors that have already been approved for other uses. So since these drugs are already approved, this method of repurposing drugs is an efficient means for antiviral drug discovery. So we tested all of these inhibitors and drugs against JC polyomavirus to see which drugs, if any, were able to reduce JC polyomavirus infection in our cell culture model. So it turns out about 40 of the drugs preliminarily were able to reduce JC infection. So we then broke those 40 drug hits into categories illustrated in this pie chart in panel 4. One of the categories that stood out to us was calcium signaling drugs, since our lab already had preliminary evidence from other experiments to support the involvement of calcium during JC infection. And finally, in the schematic shown in panel 5, you can broadly see how these calcium signaling related drugs might work to inhibit JC polyomavirus infection. We're currently working to further validate and characterize some of these calcium related drug hits from the screen to identify how these drugs might be reducing JC infection. So once we identify one or more than one of these drugs that continues to reduce JC polyomavirus infection in our experiments, it is likely that we could use said drugs as treatment for JC infection and PML. And so since these drugs are already approved for other uses, this drug screen approach is again an efficient way to discover therapeutics for viral infection. Thank you.
So we are now ready to start the final session of the day, the much anticipated award ceremony. It's my great pleasure to introduce the President of Student Government, Harrison Ransley, to start the award ceremony.
Good afternoon, everybody. It's great to be here. Student government is excited to be a major part of this event, and I'd also just like to personally thank everyone for being here today. These events are just very important, especially nowadays. And I just love to see the teamwork and effort everyone has done here, not just for themselves, but for the people and organizations around them. Each presentation was reviewed and scored by 3 independent judges, including members of the UMaine community, faculty, staff, graduate students, as well as judges external to campus and local industries, including our sponsors. Although all presentations were extremely well done and the competition was very close, the following student groups rose to the top and are recognized next. I would like to invite Executive Vice President for Academic Affairs and Provost, John Vaughn, to announce the Center for Undergraduate Research Fellowship and Maine Space Grant Consortium Research Experience Awards for summer 2021.
Great, thank you, Harrison, appreciate that. I just want to say, Harrison, it's been really wonderful working with you this last year in your role as the president of the student government. And I just want to give a shout out to the University Singers. That was absolutely amazing. I think I spent my whole afternoon listening to you. So thank you so much for that. So it is my pleasure to give out the following awards. This is the first are for the Maine Space Grant Consortium Research Experience Awards, and there's 10 of them. And so these 10 students I'll name, and I'll give their faculty mentors as well. So the first student is Jocelyn Cooper, and her faculty mentor is Dr. Rhiannon Waller. Next student is Maxwell Burtis, with the faculty mentor is Dr. Seth Campbell. Third student is Morgan Oler, who is mentored by Dr. Jean McCrae. The fourth student is Jessica Hutchinson, and that's with a mentor is Dr. Stephanie Burnett. Fifth is Sarah Losell, and their mentor is Dr. Jared Talbot. The sixth is Michael Keaty, and Dr. Seth Campbell is his mentor. In fact, the next two are also with Dr. Seth Campbell. That's Emily Holt and Jordan Farnsworth. Next is Leah Savage, and Dr. Amelia Coterre-Bou is her faculty mentor. And last but not least is Leah Parrish, and that's with Dr. Nimisha Rana Singh. So congratulations to all those Maine State Space Grant Consortium winners. Next up are 6 awardees for the Cougar Summer Fellowships. And that is, the first is Samuel Roberts. Faculty mentor is Dr. Yongjung Jang. The next student is Eileen Lieber Bendix, whose faculty mentor is Dr. Shannon McCoy. The third is Emily McLaughlin, and her faculty mentor is Dr. Edward Bernard. The next student, Cougar Summer Fellowship, goes to Rosemary Thayer, and that's with Dr. Alessio Mortaletti. Then it's Aldous Hoffman, and again with Dr. Jang. And finally, Basil White with Dr. Andre Khalil. So congratulations to all of you. Those are really, as Harrison was saying, really wonderful awards and well-deserved. It's now my pleasure to introduce Dr. Tammy Crosby, who is the Special Assistant to the Vice President for Research and Dean of the Graduate School, to name the awardees of the Susan J. Hunter Research Impact Award, the Faculty Mentor Impact Awards, and the Epic Discovery Race winners. Tammy?
Thank you, Provost Rowland. It's great to be here. Thank you for inviting me to announce these awards. These are very important. And I'm going to start with the Susan J. Hunter Presidential Research Impact Award. This award is given to one outstanding undergraduate and one graduate presenter at the UMaine Student Symposium whose project best exemplifies the University of Maine mission of teaching, research, and service by impacting Maine communities, citizens, and business. The winners are selected from nominations submitted by their faculty mentors. This prestigious award comes with a $1,000 cash prize. So this year, in the Graduate Scholar category, the award goes to Christine Hale for her symposium project number 1045. I'm going to try to pronounce this title. It's Nociceptive Sensitization in Larval and Adult Drosophila. I think I mispronounced something there, but Her work aims to combat the national opiate overuse and overdose epidemic by identifying a new biochemical pathway used by the nervous system to modulate pain sensitivity, providing the potential of the development of dozens of new, less addictive pain medications. Christine is a doctoral student in the UMaine Graduate School of Biomedical Science and Engineering, doing her doctoral work under the direction of Dr. Jeffrey Ganter, Professor of Biology at the University of New England, an institution associated with the Graduate School of Biomedical Science and Engineering. Congratulations, Christine. In the undergraduate scholar category, the award goes to Savannah Mishu for her symposium project number 708, the relationship between adverse childhood experiences, working memory, and perceived stress in older adults. Savannah's work was done in the UMaine Cognitive Aging Resiliency Enhancement Lab, also known as the CARE Lab, which is directed by her faculty mentor, Dr. Rebecca McCauley, Assistant Professor of Psychology. This project was a team effort along with students Amy Halpin and Angela Bove. So congratulations, Savannah. And check out their presentations. They're recorded online. They both did very good presentations recorded. Now I will move on to the winners of the first ever Maine Impact Epic Discovery Race. Students completed virtual challenges to discover and share their research, scholarly, and creative experiences at UMaine and UMaine Machias. Everyone that participated in the race will receive a Maine Impact Week t-shirt, like the one I'm wearing here, and the top 3 teams win a monetary award. So the first place team with a score of 2,525 points is the Splice Girls. Second place with a score of 2,513 is Team 2 Oak 3. And the third place team, Coconut Oil, with a score of 2,507. Congratulations to these teams and to all the players. I hope you enjoyed the race. It was a close race the whole week. It was a whole week-long race. And last but not least, the Faculty Mentor Impact Awards. The faculty selected for these awards were nominated by their students. We receive hundreds of nominations every year for these awards. Now more than ever, it is important to recognize the dedication of our faculty in making a difference in their students' lives. I will announce the winners by college. So in the College of Liberal Arts and Sciences, Dr. Shannon McCoy, Associate Professor of Psychology, Dr. Brian Frederick, Associate Professor of Chemistry, and Captain James Sedley, Executive Director of the School of Policy and International Affairs. In the College of Education and Human Development, Dr. Kathleen Gillen, Assistant Professor of Higher Education. In the College of Engineering, Dr. Aaron Gallant, Assistant Professor of Geotechnical Engineering, and Dr. Carissa Tilbury, Assistant Professor of Bioengineering. In the Maine Business School, Dr. Tim Liu, Associate Professor of Business Analytics. And lastly, in the College of Natural Sciences, Forestry and Agriculture, Dr. Christina Kamen, Assistant Professor of Marine Mammal Science, Dr. Pauline Kameth, Assistant Professor of Animal Health, and Dr. Liz DePoy, Professor of Social Work. So that's it for the faculty mentors. And you can check out videos of their nominations on the Maine Impact Week website. And now the graduate student government has some additional awards to present. The Vice President of the Graduate Student Government will announce these awards.
Thank you, Tammy. My name is Surya Prakash. I'm the VP of Graduate Student Government. The Graduate Student Government is extremely glad to support the UMaine Student Symposium, and today we'll be presenting the Provost Innovative and Creative Teaching Award, the Dean of Graduate School Undergraduate Mentoring Award, and the Foster Innovation Center's Student Innovation and Commercialization Awards. The Provost Innovative and Teaching Award is an award that's given to a student for the use of innovative teaching techniques, and in the way they bring the subject matter to life. The Dean of the Graduate School Undergraduate Mentoring Award is an award that's given to a graduate student who has gone above and beyond to mentor graduate students in a research setting. The Student Innovation and Commercialization Award is presented by the Foster Innovation Center The intent of this award is to recognize the research of graduate students at the University of Maine that shows promise to develop into a commercial product or service. This award is meant to showcase those students whose research has potential to generate feasible and innovative products or service. For the Provost Innovative and Creative Teaching Award, this is a tie. Michael Clay and Komla Shavana has gotten this award. For the Dean of the Undergraduate School, Graduate School Undergraduate Mentoring Award, it is also a tie. Michael Bilchik and Shelby Helvig has gotten this award. For Foster Innovation Center Student Innovation and Commercialization Award, the first place goes to Natalie Van Adenker, and the second place goes to myself, and the third place goes to Taka Alasadi. Congratulations to all the awardees today. I would like to invite the Associate Vice President for Research, Dr. Ali Abedi, and Associate Vice President for Graduate Studies, Scott Delcutt, to join me to announce these student awardees. Thank you.
Thank you, Saria, and congratulations again to everyone. We are now moving into the UMaine Student Symposium Awards, which are presented to one undergraduate and one graduate project in each of the following 10 technical categories. The selection has been made based on the published rubric and evaluated by 3 judges. Each project will receive $500 cash prize as we move forward through the presentation. So Scott, you can
Sorry, I have to unmute. Thank you, Dr. Abedi. I'm pleased to start with announcing the graduate student winner in the first category of Allied Health. The winner is Rocky Coastlines, advised by Dr. Jennifer Crittenden.
And the undergraduate project winner in the same category of Allied Health is Elena Kolesnikova, advised by Dr. Valerie Herbert. The next category is arts.
And the graduate winner in the category of arts is Anna Soul, advised by Dr. Owen Smith.
And the undergraduate winner in the arts category is Caitlin Sharples, advised by Dr. John Ippolito.
The next category is biomedical sciences, and I'm pleased to announce the graduate winner is Avery Bond, advised by Dr. Melissa McGinnis.
And the undergraduate winner for the biomedical sciences category is Lydia Curran, advised by Dr. Len Cass. We didn't have any graduate submission in the business category, so we are announcing the undergraduate project winner for the business category, and that goes to Jaslyn Thomas, advised by Dr. Stefano Tijerina.
In the next category of education, the graduate recipient of the prize is Camden Bach, advised by Dr. Justin Dimmel.
And the undergraduate awardee in the education category goes to Tom Adams, advised by Dr. Rebecca Buchanan. The next category is engineering and information sciences.
And in this category, the graduate winner is Dan Regan, advised by Dr. Caitlin Howell.
And the undergraduate project winner in engineering and information sciences goes to Josh Hamilton, advised by Dr. Carissa Tilbury.
The next category is interdisciplinary research, and the graduate winner in this category is Sahar Ruzbahani, advised by Dr. Michael Mason.
And the undergraduate project winner in the interdisciplinary research category is Nicholas Sherman, advised by Michael Scott. The next category is natural sciences.
And the graduate winner in natural sciences is Griffin Dill, advised by Dr. Allison Gardner.
And the undergraduate awardee in the natural sciences category is Anna Briley, advised by Dr. Nishad Jayasundra.
In the category of physical and mathematical sciences, the graduate recipient is Pradnya Rao, advised by Dr. Carl Tripp.
And the undergraduate winner for physical and mathematical sciences is Peng Cheng, advised by Dr. William Gramlich. And the next category is social sciences and humanities.
And the graduate winner in social sciences and humanities is Morgan Stosick, advised by Dr. Molly Rubin.
And the undergraduate project winner for the social sciences and humanities is Erin Dustin, advised by Dr. Jordan LeBouf. I would like to thank all the award winners again, and thank you, Harrison. Back to you.
All right, thank you very much. Congratulations to all the student presenters and awardees one more time. You know, you all did an amazing job, and I was personally just blown away by the work that you all are doing. I would also like to thank the faculty advisors who have provided these students with the opportunity to just conduct this research. I know that we students greatly appreciate the research opportunities that we come across and that you continue to provide with. I would also like to thank the judges and the 40+ planning operation volunteers who have helped make today possible. This event continues to grow each year. It would not be possible to put on such an event without you all. I'd also like to give a huge round of applause to our sponsors. You provide us students with this presentation platform. Many of us, this is the first time we get to showcase our hard work. And just to go through all of our sponsors again, the hosts of this event were Student Government, the Graduate Student Government, Center for Undergraduate Research, and the Office of the Vice President for Research and Dean of the Graduate School. We also have different levels of sponsors, with the first ones being diamond sponsors. And the sponsor that falls into that category is Texas Instruments. Up next, we have our platinum sponsor of Maine Technology Institute. The next round of sponsors are our gold sponsors. They're Activas Diagnostics, IEEE Maine Section, and Northern Light Health. And then our silver sponsors are as follows: Allied Engineering, the Jackson Lab, Landry Construction, President Joanne Farini Mundy and Richard Mundy, ND Paper, Power Engineers, SMRT Architects and Engineers, St. Joseph Care, TRC Companies, Inc., University Credit Union, the Upstart Center for Entrepreneurship. This concludes our 2021 Student Symposium. Thank you to all of the presenters, and thank you all for attending. Fill out our online survey to help us improve this event, and please join us again on April 15th, 2022 for our next event.